This lecture we're going to discuss microbial genetics. This lecture is going to be a little longer than the other ones simply because the material is complex and far-reaching. The concepts are essential to microbiology, and it may be that you'll need to listen to this lecture a few times, but you will definitely get great rewards from understanding the material very well. So we're going to start off the discussion by bringing back some important definitions, the first of which is what is a gene. And there have been several changes in the definition of a gene. Most recently, a gene now is thought of as a section of the chromosome, or a section actually of DNA, that encodes the primary sequence of some final gene product, which can be either a polypeptide or RNA. But initially, back in the 60s, a gene was thought of to be a segment of genetic material that encoded for one enzyme. The theory was called one gene, one enzyme hypothesis. Then we broadened the definition to include one gene, one polypeptide. And now we've also included RNA as part of the definition. So genes can also include regulatory sequences, and these are parts of the DNA that are actually there to regulate the transcription of other genes, and these sequences provide signals that denote the beginning or the end or some kind of influence of the transcription process itself, or the regulatory sequence functions as a place where transcription is begun, or a place where replication occurs, or where recombination occurs. So it's also been broadened to include several different things, but basically the true underlying function is in some kind of way it regulates transcription. So the genetic code, we've alluded to this genetic code in a couple of our discussions already, and today we're actually going to clearly define what it means. So in the DNA, the way it works is that each DNA sequence and each subsequent RNA sequence encodes for one amino acid, and generally it takes three base pairs to encode for one amino acid, and depending on the order of arrangement of these nucleotide bases, that will determine the arrangement of the ultimate arrangement of these amino acid sequences. And each of these amino acids are joined by a peptide bond, and depending on which amino acid actually occurs in these various positions will also influence how the protein is shaped after post-translation. So transcription and replication, as we discussed in earlier lectures, is basically very similar to transcription and replication in eukaryotes. So bacterial transcription and replication, as I said, is very similar to the eukaryotic processes. So we have transcription and translation occurring here, and we have recombination and replication occurring here. But the thing that is very different about bacteria that we're going to see in the next couple of panels is we can also have gene transfer occurring between individuals of the same generation. So we certainly get inheritance of the parental DNA into the daughter cells, but we can also exchange genetic material between the daughter cells, and that's actually what makes microbial genetics so complex. So here we have replication. Okay, remember that word, replication. Of bacterial DNA is very similar to replication of eukaryotic DNA. Basically, we need to copy both sides of the DNA strand. So we get this unfolding or unwinding of the DNA, and we have this replication fork, and then we actually have a copying of both sides of the DNA in the circular motion, and then we basically flip the circles, and then we have a complete copy of the DNA in the daughter cell. Now, DNA transcription in bacteria also work rather similarly to eukaryotic cells, and that's where the DNA ultimately produces proteins, right, according to the arrangement of the nucleotide bases where we talked about in the genetic code slide. So we take DNA and it gets transcribed to RNA, and that RNA polymerase binds to the promoter sequence. We get transcription beginning, and then it stops when we reach that stop codon or that terminator sequence. So here is a picture of bacterial transcription. So you can see over here This is very similar to eukaryotic transcription and translation. However, there is a major difference. What is that difference? There is no nucleus, okay? So all this needs to go on in the cytoplasm. And, biochemically it's different, of course, but in terms of functionally, it's not really that much different at all. So here we have another slide of the actual process of bacterial transcription. So you can see the RNA molecule is assembling these nucleotides to match up in a complementary way with the information on the DNA strand. And the synthesis, it moves along the DNA, and then the RNA kind of comes off in the opposite direction, and it moves, the transcription machinery moves in one direction and the RNA moves in the other. So then we get translation. What is translation? Remember, translation is assembling the amino acids into the protein based on the codes that were transcribed by the messenger RNA. So messenger RNA is translated into codons. Remember those 3 nucleotides? And the messenger RNA, starts, and then it stops based on that regulatory sequence. So the protein in a bacteria is used for cell metabolism and growth. So gene regulation, how can we regulate all of those processes? We certainly have different kind of genes. So we have the constitutive genes that are just expressed at a fixed rate. They're just continually made at some rate that's fixed. But other genes are expressed only when they are needed, and those genes are called repressible genes and inducible genes. And we're going to have a little in-depth discussion about what that means just right after this slide. So regulation of transcription is a way in which we can figure out and initiate or stop sequencing, or actually not sequencing, but synthesizing certain proteins. So for instance, if you have a bacteria that produces a toxin, right, when it infects a organism, a human host or an animal host, then that toxin requires the production of potentially some sort of protein substance, and that regulatory sequence on that bacterial DNA will need to get initiated in order for the toxin to get produced. So we have this regulatory gene here, sequence or portion of the DNA. We have this control region, and then we actually have the region of the DNA that's encoded for certain structural proteins. So in terms of repression, we have the regulatory gene actually produces this repressor protein that then lands on the DNA and essentially stops the transcription from occurring. So the transcription starts, for instance, and with this repressor protein, it just stops right there, and that protein does not get synthesized. And so what protein does not get synthesized is the protein out here. So that's one way that we can actually cause repression or stopping of the synthesis of a particular protein. Now, in terms of induction of that, we can actually have it occur in a couple of ways. So we can either have a secondary molecule called an inducer that will land on the repressor protein and actually inactivate it. So that's one way. Another way, there's other regions on the DNA that are actually set up that if something lands on that DNA, that will cause the machinery to start. But in this case, we actually stop the repression, and then we get transcription and translation, and then ultimately we get the protein gets produced. And in the case of that bacteria, we get that toxin. Okay, we get that toxin produced by taking away the function of that repressor protein. Now, let's talk about mutation. Now, mutation is another concept that's common to both eukaryotic and prokaryotic cells. And the question, again, has arisen for quite a long time in genetics in terms of defining what a mutation is. So Eric Pianka, who was a very famous ecologist, defined a mutation as a genetic change to a germ cell. Now, what is a germ cell? When a eukaryotic cell, a germ cell, or in a human host, a germ cell is an ova or a sperm. And when we have a genetic change to a germ cell, that genetic change is passed on to our offspring. And so in terms of evolutionary biology, that genetic change will either enhance or disrupt a population's fitness to reproduce. So this has an evolutionary connotation. But, certainly these concepts apply to bacteria as well, especially in the case of the antibiotic resistance that we discussed in the beginning of the course. So only germline mutations are transmitted to offspring. Maureen Curry, who's another famous geneticist, clarified that terminology. Now, there are also some definitions of genetic mutation in relationship to molecular biology. And a genetic mutation is, in molecular terms, a permanent transmissible change in the nucleotide sequence of a chromosome, usually in a single gene, that leads to a change or loss of its normal function. And again, if we have that change occurring in germ cells, that change is transmitted to all the cells in the organism. Now, the other concept about mutations that's important is that mutations are rare, and also they're random. Now, these mutations can be beneficial, neutral, or harmful. And in the respect that they're random, it doesn't matter if the mutation is useful or not. The fact that it occurs at all, it's a random process. Not all mutations matter. Some, if they're integral to particular functioning of some protein, then they do matter, but they can occur in a part of the DNA that's not a gene or not expressed in that particular manner. And so in that case, not all mutations matter. Somatic mutations, now we were just talking about germline mutations. Now, somatic cell mutations are mutations that occur in cells that are not germ cells. They're not reproductive cells, and they won't pass on to our offspring. So, for instance, if we stay out too long in the sun, our skin cells may have a mutation that's induced in that particular area that got sunburned, but the rest of our body isn't going to have that mutation, just that part of the body that was exposed to too much sun. So the example here, the golden color of this apple was caused by a somatic mutation, but the seeds of the apple, so if we planted this apple, if we planted the seeds, the apple would not grow into being multicolored. It was just that particular apple had that particular genetic change. Now, these mutations are different, again, from germline mutations, because germline mutations, all of the cells in our body will carry those germline mutations.